So it is 3 o'clock now. We'll take a couple minutes. I still see the participants list is slowly growing. So we'll just get started off with a brief introduction on Zoom. So welcome everyone to the intro to the IU Herbarium photo hunt. So we've got a few people here to help you be a citizen scientist. But before we can do that, we want to make sure that you are comfortable here at the meeting. If you look at the bottom of your screen, you should have the options for the chat, right? So it looks a little word bubble there. You can click on that and open up your chat so you can log your questions as they come to you. Or if you want to just throw out a thumbs up or a smiley face, there's different reactions that you can play around with, because I know as a presenter myself, it is definitely a new world where I am presenting to seemingly no one. So we do appreciate a thumbs up to every once in a while if you like something, or if you just to make sure that you're still out there and you can still hear us and everything's good. So thank you again for being here this afternoon. It should be a great program where Purdue and IU are partnering to help the Herbarium's photo archives. So again, this program will be recorded. Please keep your mics and your cameras off. The cameras off will help with the streaming for people with low internet, especially, or poor internet. Keeping the cameras off will help with that. And keep your mics muted, but we will, or potentially have the time for some Q&A at the end. So if we can unmute at that time to ask your questions for Paul or John or Eric. And so with that, I don't see any questions popping up into the chat at the moment. Got a thumbs up from John, so I know you guys can hear me out there. Welcome again. I'm Karen Mitchell. I'm the Ag and Natural Resources Educator in Tippecanoe County, and here today with a few people from IU to talk about the IU Herbarium. But before we get into that, why are we making this partnership? Why, you don't see Purdue and IU working together a whole lot, but as much as you may not see it, we do work together. And now we're trying to pull in our volunteers to really leverage all of our resources to build a great archive of photos. And so the importance of citizen scientists, you're the eyes and ears that are all around Indiana. So the university researchers can't be everywhere across Indiana looking at every nook and cranny, but I know there's master gardeners and other volunteers that are out there. And so using your smartphone, sharing this information that you see, it's expanding the university research's abilities to reach every nook and cranny across Indiana. So it's helping move forward the university's research, but it's also helping to build that community network, right? So getting out there, talking with your nature preserves that are in the area, really getting to know your neighbors or the nearby farmers or the woodland owners. So it's helping to build that community in your local areas. And not only is it important to build that community, but also develop more of a personal connection, so making sure that you know what's out there. You may have a natural preserve right around the corner that you've never visited. So this is maybe an opportunity or a motivation to get out there and visit some places you haven't and share what you see and learn. But as master gardeners, I know you are all eager to get out there and get some volunteer hours. And so this is providing that opportunity while social distancing, and maybe doing it with the whole family. So bringing the kids out and learning to identify some native species out there. So it's a really important partnership that we've developed here. And I'm really happy to see, Purdue Master Gardeners helping out the IU Herbarium, because it is a great resource. But with that, we'll move on to introductions of Eric and Paul. So Paul Rothrock is the Associate Curator for the Indiana University of IU Herbarium. And then we have Eric Knox as the Director of the IU Herbarium. And then all of us know John Ork, our State Coordinator for the Purdue Master Gardener Program. And then I'm Karen Mitchell, the Ag Educator in Tippecanoe County. So happy to be here and happy to have Paul, Eric, and John with us as well to share the importance of this great partnership and how to do it. So with that, I will go ahead and pass it on to Eric, if you are ready. Participant list has climbed a little bit during my introduction, but it's pretty slowed down pretty much. So let's go ahead and get started with yours, Eric.
Okay. So I'm going to go ahead and share my screen. And we'll put it up there, and Paul will lead off.
There we go. Yes, this is the voice of Paul Rothrock, as opposed to my colleague in crime, which is the more bass voice of Eric. And over the last 5 years, we have been creating a botanical information system. Basically, information about all of the species of plants that grow spontaneously in the state of Indiana. And in that database portal, we have live photographs of live plants, but only about half of the species have been documented in that manner. And so this project is designed to encourage all of you to participate in populating the website with the other photos that we need. So if you, go ahead, Eric.
So here is the URL for the photographic scavenger hunt project, but the message that John sent out to everybody on March 10th has hyperlinks to the project website, to the Consortium of Midwest Herbaria data portal, which we'll show you a bit about, to the IU Herbarium webpage and stuff. So that email message from John contains hyperlinks to all of the things that you will see today.
So this is our project website, Photographic Scavenger Hunt. And I love our caption there, pull on your boots and grab your camera. This is the time of year when we all want to overcome our case of cabin fever and get out and see some of the beautiful plants that we find in our Indiana landscape. We're going to take you through this website by following the items that are on the navigation bar that begins with hunting lists and so on.
So here's where.
So we have, since this is a scavenger hunt, we need to tell you kind of, well, what we're looking for. And so we have created a series of hunting lists that detail the species that are needed for our purposes. This map of Indiana shows, though, that we've divided that hunting list down into regions of the state, or ecoregions. And we happen to be in the Highland Rim ecoregion here in Bloomington, but some of you are probably over in the Bluegrass region in the southeastern part of the state. And you have different species of plants that you can find than are here in our Highland Rim area. And certainly that's the case if you get up into that northwestern morainal area along Lake Michigan, a very different set of plant species up there. So if you need to find out which ecoregion you are in and can't quite tell from the map, click on that alphabetical list of counties and you'll find there your county and your ecoregion. So here's the page a little further down that same page with the buttons or the links to the hunt lists for each of the ecoregions. We have bluegrass and north. Wait, Eric, I only see 8. I thought there were 9 ecoregions in the state.
Well, the Tipton-Till Plain region didn't fit on the screenshot, but when I pushed the button for the Tipton-Till Plain, it sent directly to my download folder a spreadsheet for the spring Tipton-Till Plain listing that includes the family, the scientific name, when we have records of them being in flower from the IU Herbarium, but also a photographic priority. So Paul went through and identified species that we don't have any photographs of live plants, and other ones where there are photographs that various people have put in, but they're not showing the diagnostic features that really allow us to distinguish one species from another close species. So we've sort of graded things as far as priority, where the high priority ones we really want, but we also want the medium priority ones if they're better than what is already up there. In a few cases, like for Cerastum fontanum, it's been collected in this region, both in March and in April. Spring's a little bit late this year, so some of the things that have been collected in March before, yep, they're still going to be there in April, but come down here to Dirca palustris, we only have a specimen that was collected in March, but it'll probably still be flowering in April, given how late the spring is this year.
Mm-hmm. Moving to the second item on our navigation bar, that's we had a species hunt list, but how do you find out about the species? Where are they and what do they look like? So for this, the information is available on our data portal called the midwestherbaria.org website. And we want to show you some of the features of this website and how to gather the information about these species from the website.
So if you scroll farther down that page, there are 4 buttons here, and pushing each one of these will pull up an information sheet that helps guide you when you actually use the Consortium of Midwest Herbaria website, and there was a link for it up there. So if I start by pushing species pages, it takes me to information about species pages.
And this one happens to be a species called Abutylon theophrasti. Oh, let's cease with that scientific name stuff. People don't want to hear those all the time. This is also known as velvet leaf. So at number 2, we have common names such as velvet leaf, and there may be multiple common names that we are aware of. Plus, there may be synonyms within the scientific names as well. Then below that at 3 is a banner photo that shows the plant. And underneath each photo is the name of the photographer. So
who took that picture, Paul?
I did. Isn't that a beauty?
It is a beauty.
So if you take a picture that we use, you will have full credit underneath your photo. Beneath the banner photo are 4 additional photos which show diagnostic features of the plant. So I notice that one of them gives a closer view of the flower. Another one is giving you a closer view of the leaf shape. Up at item 4, this is the printed information. Some of the tabs have full-blown descriptions of the plant, but we've opened the tab to the Indiana Flora tab. In this one, we have all of the notes from Charlie Deem's 1940 Flora of Indiana. These notes tend to be more ecological in nature and more folksy, and I think you would enjoy just kind of reading through some of those and seeing what wisdom he brought to that particular volume. Below that, I'm gradually adding some additional information about each of the species. What kind of conservatism does it have? In other words, how much does it tolerate human disturbance? What kind of wetness is the habitat in which it grows? Is it a wetland habitat or a drier habitat? And then the diagnostic features that you key in on, you really focus in on if you're trying to identify that particular species as velvet leaf. And then item number 5, this is a link to a mapping function that shows you where in Indiana we have georeferenced specimens that have been fully located by latitude and longitude, and you'll map them out across the state of Indiana.
So the overall database has many specimens. Here's where at the IU herbarium, we've georeferenced all of our specimens, but for other herbaria, it's still a work in progress. So oftentimes they've got entries for them, but they still haven't done that final work of identifying where exactly the collecting site was. And so the map is only going to show you that subset that has the latitude and longitude included.
Now, how to get to a species page? Well, on the home page of midwestherbarium.org is a search window entitled Search Taxon.
What does taxon mean?
Yeah, taxon means basically the species name. and you type in the taxon name, the species name into that window and hit the search button.
But could I also cut and paste it from one of the hunting lists to get there?
That's a great idea, Eric. That would save having to memorize the name and remember how to actually spell it. Just copy and paste it from one of the hunt lists and get to that species that you want to see.
So what's important for people to know is that sitting behind this is a very extensive database of the names. And so here's where only names that have already been entered will appear there. So as you start typing, it's going to have a drop-down list where, in relatively few characters, you can get the genus, and then in relatively few characters, you can get the species and stuff. But it only works with scientific names. We'll show you how to get to places using common names in just a moment. So we're now going to hit the button searching the Consortium of Midwest Herbaria database, and it pulls up a different help sheet.
Yeah, so you want to find out, okay, well, where does a species on our hunt list occur in my area? You can use this search function, the specimen search function. As you click on it, it opens a drop-down window that includes search collections. So clicking on that will bring you to a page with a list of over 400 herbaria that are part of a consortium of herbaria that are creating a massive database. We want to specifically look at, though, the Indiana University collection because it's very specific to Indiana. And as Eric has just indicated, it's very complete in the effort to get it all databased and geo-referenced.
And so one way of thinking about it is that these 400 plus herbaria, it's like having a shopping mall with 400 different stores in it. But then there are different data portals, different entryways to get into that big shopping complex. And the Consortium of Midwest Herbaria is just one of those ways of getting in. And as we'll show you, there are some things that are only relevant to us here in the Midwest. And so these different portals have slightly different properties, but they all lead to the same 400 plus herbaria. But if you don't want to search all 400 plus herbaria, uncheck the box to deselect all, and they'll all disappear. And then scroll down to where you get Indiana University Herbarium and check that box. There's also another herbarium down at IU Southeast, which has a collection of about 10,000 specimens. But then go ahead and hit the search button, which will take you to a search page that has lots of different criteria that you can use to search for things.
So in this example, we're going to try to do a search on the species Viola lanceolata. It's one of our woodland violets. It's fairly uncommon in the state, but we'll find out just in a moment. Let's search on it. So if you start typing in Viola lanceolata in the scientific name box, you soon will get a drop-down window that shows not only that, but also some of the subspecies and varieties that have been used for this species. But let's stay with the broader Viola lanceolata, and then go up to number 2 and hit the list display button. This will open then a occurrence record page, which it echoes back to the input that you have put in, Viola lanceolata, and using the IU Herbarium collection. If you look it over to number 2, this indicates that for Indiana, we have 27 records. So as indicated, this is a fairly uncommon species of violet, mostly in the northwestern part of the state. At number 3 are each of the individual specimen records in summary, but you can click on the full record details to see all of the information as to when and where it was collected. But also over to the right, you could click on that image, and that would give you an enlargement of the actual preserved specimen that we have in the herbarium. And those images have enough resolution that you can almost see hairs on the leaves. I mean, they're really quite detailed. You notice that for the viola, since this was a viola lanceolata in a broad sense, we have both subspecies Vittata and subspecies lanceolata in the list of specimens that have been found in this particular search.
So if we had typed in Viola lanceolata, subspecies Vittata, we would have only picked up the specimens for that subspecies, whereas if we had put in Viola lanceolata, subspecies lanceolata, we would have only picked up the records for that subspecies.
On that same page in the upper right-hand corner, there are several other buttons of value. Number one allows you to change the view to a table view. Number two is a download button. So all the information that is in our botanical information system, in this data portal, can be downloaded as spreadsheets to your computer for manipulation and use in other forms. There are several additional tabs. This one that says Maps, if you click specifically on the word Maps, that will open up a Google map that shows, once again, the georeferenced specimens. So for us, all of ours are georeferenced, and those will show up on a map of Indiana.
And each of the dots that shows up is a live link to the actual specimen page. So you can see what the distribution is and then click on a dot, and it will take you directly to the corresponding specimen page.
So just to review, all of this then is to say, you got a hunt list. Now, how to find out where these things have grown in the past and where you might go looking for them. This gives you a sense of how to search for that information within the Consortium of Midwest Herbarium.
So if we go to working with species checklists, it opens up another information page here.
So under that Flora Projects button or tab on the navigation bar, click on the state of Indiana, and we'll open a page that has a list of checklists that are specific sites in Indiana. For example, we do have one for Monroe County in particular, but also checklists for those 9 ecoregions that were on the map at the beginning of our program. So if we follow with the Bluffton till plain, let's click on that particular ecoregion and see what a checklist looks like.
But before we leave here, here's the golden key symbol.
You know, I missed that.
We could go directly to the golden key from here, which we'll show you in a moment.
That sounds like something really secret. Wow. Okay. Shh, don't tell anybody. All right. So here's the Bluffton till plain ecoregion checklist. At number 1, it gives us the details as to the counties that are included in that ecoregion. Number 2 is the list of species. We have both the scientific name in blue and the common name in black. So there we have, once again, our friend Abutilon theophrasti, or velvet leaf. Up at number 3, Eric, that's the golden key again.
It's the golden key. Yeah, for some reason, the symbol went from being a golden key to looking a little bit more like a skeleton key. But from in here, you can get to the golden key. We'll show you what the golden key is.
Okay, keep them all waiting. Number 4 is a filter panel where you can change the format of the checklist. So for example, right now, it is set up to show the species alphabetized by genus and species, but you could change it to be alphabetized by plant family. Or you could click on that display as images and then build the list, and it will show a gallery of photos. From each species page, it will take the banner photo and it'll lay it out as an array on that page.
But here's where you could also search for a subset. And so if you wanted to look for oaks, you could put in common names and put oak, and anything that has oak in the common name will show up in the list here and get rid of everything else. Here's where the genus name for the oaks is Quercus. So if you know that, you can search for Quercus and it'll take you only to Quercus. And then you could have the images just for the oak species. So there are lots of things that you can do with these checklists. But now,
finally, Eric,
the pièce de résistance, how to identify species using the golden key.
The golden key.
Everybody say this with us. The golden
Golden
key.
key.
I heard you all at home there. So if we click on that button, it explains how the golden key works.
Yeah, so this is often the big bugaboo for citizen scientists is how to identify a species of plant or animal, what have you. And this is a rather useful tool because it starts off by narrowing the list of everything down by ecoregion. And from there, we have chosen the bluegrass ecoregion with 465 species, but the state of Indiana has a total of over 2,700 species. So we've already eliminated about half of the species in our effort to identify an unknown by just choosing the ecoregion. Then on the left-hand column, we have a series of plant traits, and we made an effort to choose things that are fairly simple, as simple as we can. And on the right hand, it has a list of candidate species that we're going to continually narrow down by checking off individual traits.
So as an example, if we take false rue anemone, which will be flowering soon, so this is Anemone bitornatum. Some of you may be familiar with it, knowing that it's an herbaceous plant with basal and alternate compound leaves, white flowers that only have one whorl of colored parts, happen to be sepals, not petals, but that's not obvious. There are numerous stamens, multiple ovaries that form fruits called follicles, and it blooms early in spring. So here's where, if we check that it's an herbaceous plant, we get rid of all the ones that are not herbaceous, so we're down to 1183, but it will also then pull up additional characters because the way that the character list works is that for whatever species are still here, it looks for characters that have a high discriminant value. And so indicating that it has blade division, some or all being fully compound, reduces the number down to 165 in the bluegrass region. And if we pick that the leaves are basal, then we're down to 32 species. We pick that white is the principal flower color, we're down to 19, and determining that there's only one whorl of flower parts, right? So the perianth consists of only one whorl.
What's
then we're down to 3.
Perianth sounds like some kind of an insect. What's a perianth?
Well, the perianth is a general term that refers to sepals or petals or either one of those in combination. And in this case, it's really hard to tell whether they're sepals or petals. It's only through developmental studies that we really know that they're sepals that are now white and not petals. So perianth is a generic term. And here's where there are information buttons that will take you to Wikipedia. So if you're not familiar with this word, you can learn as you go. Here's where we're now down to 3 species that are in the Ranunculaceae. And each one of these is a live link that would take you to the species page for it. But a couple of the developments that are in progress with the person who designed the underlying program that we use called Symbiota, a man by the name of Ed Gilbert, who's in Arizona, we're going to have it automatically convert into a photo gallery when we get down to a certain number, so you don't have to go individually to each of these species pages. We'll see the top image for it. And we're also going to change the links from Wikipedia to an internal illustrated glossary, because Wikipedia tends to be word heavy and you try to look up one word and then it has 5 more words that you've never seen before. So here's where we're going to use the photographs both to illustrate the species pages, but also looking for good examples that illustrate the various features that are used in the golden key.
The next item in our project website is some tips on how to take good photos of plants. And the beginning point is really the most important one, and that is, while you're there, take lots of photos. Pretend you're doing a photo shoot of some glamorous model, and you're trying to get all the different angles and different lighting, different parts, get close-ups of the leaves, get the backside of the flower, get the habit of the plant. So take lots of photos because the electrons are cheap. You don't have to develop film anymore. Secondly, you may visit the plant when it's in flower, but it may not have fruit. And so it's valuable to go back and revisit the same plant or the same population at a little bit later date. Now, it just so happens in the example photo that we have, this species, tall thimbleweed, has both flower and young fruit heads on the same plant at the same time. So we were able to get both things with one image, but that's often not the case. Third item to take note of is, for our purposes, when we're trying to edit the photos, it's better to slightly underexpose an image rather than overexpose it. So going back to the same flower picture, when you have a flower that has white perianth parts, or even say yellow petals, those can easily burn out if there's a slight overexposure. But if you slightly underexpose, that information is still there, and we can enhance it with Photoshop. And we can take these darker areas maybe of the fruits and enlighten them up a little bit as needed. So we can do a lot of editing of a basically good photograph.
For those of you who watch police shows on television, and there happens to be a camera across the street that catches the back of somebody's head, and they go, oh, okay, now go ahead and rotate and enhance, and my, there's the face of the culprit. All of this is make-believe. You can't do that. So we're working with the image that you send us. But yes, things that are slightly underexposed, we can bring out the other parts without losing the nice shadowing on the petals here. But if it's overexposed, it's all going to be white, and there's no way for us to sort of work that backwards.
The next item on our navigation bar is how to submit photos. And to begin, we want you to collect information about the when and where the photograph was taken, because if we had that information, then we can make maximal use of your image. So here's an example of a photo I took last summer while on an outing with a group of other botanists, and we weren't able to at the time collect specimens, but I could get a nice diagnostic photo, and it has now been entered into the database as an observation with photo voucher instead of a physical specimen in the herbarium. When I took the picture, I knew exactly where I was. My camera file name has the date in the metadata. I was able to pinpoint the latitude and longitude. You can do that with a smartphone or Google Maps. And now that is a photo observation. So I collected all the necessary information to make the maximum value out of this particular photo.
And so herbarium specimens are still the gold standard for being able to identify plants, because we can go ahead and dissect a flower and see what's sitting inside, but good photographic observations, right, are still scientifically valid. And so if we get the information about where exactly the photograph was taken, then we have not just a photograph, but we have a scientific observation. So you really will be citizen scientists at that point.
So on the website, there's more information available about each of the items that are part of a complete reporting of the locality and information about your photo. Also, though, there's a photo submission data sheet that should accompany your photo. So if you click on that particular button, it will download a blank data sheet where you can put your photograph file name. It might be some obscure combination of letters and numbers from your camera, or perhaps you've been in the habit of renaming it with a plant name or something like that. Put your tentative species identification in the next column. Third column, your name. And this is important that you put your name in the way you wish it to appear on the website.
So do you want it to be Abraham Lincoln or Abe Lincoln? Because whatever you list as the photographer's name is how we're going to give you photo credit for it.
The next column is for the date of the photograph, and we would ask that it be in the format with 4 digits for the year, dash, 2 digits for the month, dash, and 2 digits for the day.
So this may be an unusual date format for most people, but the reason that this is the data standard is that if you sort alphanumerically, it will also sort chronologically, whereas any other date format does not do that.
The next column is for the state, and in most cases, this will be Indiana, but undoubtedly some of you live near a neighboring state and might find yourself drifting over into Ohio or Kentucky or Michigan or Illinois, and that's fine. Just make sure you have the correct state on the locality list, the county, the locality. This would be something about what you would see as you look for this on a map. So it's near Upland, Indiana, maybe a quarter mile north on State Road 26, something like that. The latitude and longitude, which you've acquired from your smartphone, and then the habitat in which that particular plant was found. Could be it's a roadside, could be a nice woodlands, dry woods, marsh, whatever it is.
So here's where for each of these columns, if you click on the little plus there, it will provide an explanation that's a little bit more detailed than we've just given you right now.
Finally, if you keep scrolling down that same page, we have the place for your online submission form, where you put in your photographs and your data sheet and send them our way. There's also on each of the pages within the website, upload photos and data sheet button on every page. So there are multiple ways to get to this particular function.
And here's where we ask that you rename the photo submission data sheet that accompanies your photographs with your last name, first name, and then the date following the standardized format. So if Abraham Lincoln submitted something on February 13th, 2021, I think it'd be a little bit old, right? It'd be Lincoln Abraham 2021-02-13. So
you're ready now to actually submit the photos. Eric, but only 30 megabytes. That's not very much for photos because that's just a couple photos at a time. There must be some secret to this.
Well, here's where if they go ahead and put it into a zipped folder, it helps shrink the overall size of the photographs that are in there. And so creating a zip folder is relatively easy. We have instructions on how to do it on a Mac and how to do it on a PC. Just hit the pluses here. It will show you in a few simple steps. But you're right. If somebody goes out and takes 80 photographs, it's not going to fit in 30 megabytes. And so here's where if you go ahead and make a zip folder and it's more than 30, it means you just have to divide it up into 2 or more folders, in which case name them your last name, first name, the date, understroke 1, understroke 2, etc. And you can upload as many as 20 such zip folders in one go.
So that way you could get up to 600 megabytes in one submission instead of just 30. All right, so there we are. Are you ready for your upload? You put your name, first name and last name, your email address, and you go to select files and you navigate to the place on your computer where you have your zipped folders and click on those. Once you have everything in place, you hit the submit button and voila, we get a notification that something has been submitted, and you in return will get a thank you back in a return email.
And so if you have multiple files that you want to send us. You select and you can pick one, right? And it'll upload it, and you can select and pick another one, right? And depending on how your computer is set up, sometimes you can select a set of them and we'll pull them all up at once and list the files so you can double check them before you hit submit. But that will send us to us, and here's where they'll come in individual folders, and we'll recompile those into one overall folder once they get through the ether to reach us.
All right, finally, last two items on our project website, frequently asked questions. And any one of these, by clicking on the plus sign, it will enlarge that and give you kind of our answer or further information. Or if those don't quite do the job, then contact us. And we have a dedicated email called photohunt@indiana.edu, or give a call during the day. Our herbarium assistant is typically there to answer the telephone, or leave a voicemail and we'll get back to you as soon as possible, because your phone call really is important to us.
So we'd like to thank the funding agencies that provided the money to hire our wonderful assistant and photographic expert par excellence, but also all of the collaborating institutions throughout Indiana, including the Purdue Extension Master Gardener Program. And with that, we'll open things up to see if there are any additional questions.
Yeah, thank you, Eric, and thank you, Paul. I think we could take, we've got a couple questions just on the project in general. And so I want to go back, and before we get to our portion of it, we'll talk about some of the guidelines for reporting these hours and activities towards volunteer hours. So we'll get to that in just a minute. So there's a couple questions here. Could you address how to find latitude and longitude on your smartphone?
So that information is on the project website. And here's where some smartphones, right, have a GPS app already on it. So it's really easy. It just picks stuff up from the satellites. You can go to Google Maps and try to figure out where exactly you are and simply click on it and it will give you the longitude and latitude. But this is explained on the project website.
Under the frequently asked questions, it's theirs for sure.
Okay, great. And let's see here. If we take multiple photos, then is the same information on the data page repeated?
And the answer there is yes. We want information for each photo because each photo should have a unique identifier. But if you're taking multiple shots of a single plant, it's really easy on a spreadsheet to just copy that information down in those columns. So it becomes very easy at that point.
Great, great. And another question here. Are you looking for pictures that are found in the wild only?
Well, we're looking for photographs of the native and naturalized species in Indiana. So those plants that are growing spontaneously, and somewhere between a quarter and a third of the species in Indiana have actually been introduced from someplace else. We call them invasive species if they're wildly out of control, but a number of things people would never know because they're relatively well-behaved species that they actually originated someplace else. So here's where the state flower is the peony, but that is cultivated. It does not grow spontaneously. So we're not looking for photographs of cultivated plants, but if you have a lot of native species in your garden, right, we would love to have good photographs because it tends to be, sunny conditions and the plants are growing well. So it's not just going out and photographing things in the wild, but rather we're looking for those species that make up the native and naturalized flora of Indiana, not cultivated species.
Yeah, I guess that's a good point. You really, oh, sorry, Paul, go ahead.
I was just going to say, in fact, a number of things that are missing could actually be rather common weeds that could be on roadsides and other less attractive places. We just don't have a photograph at this point.
Great. And I've got a question here about hours, but I'll come back to that because we're going to do our slides in just a minute. But I have natives that grow that have grown on my own personal property as well as natives I've planted. And so, yeah, you just answered that question. So that's been answered. So if Karen could share the slides, our slides, I'll finish out with some guidelines for reporting this as a Purdue Extension Master Gardener volunteer activity. And I'll preface this by saying you should always talk to your Purdue Extension Master Gardener County Coordinator and to be sure that you are reporting the correct information and that those activities are approved by your coordinator. But if Karen, you want to advance for me to the next slide, and we'll get started here. But so volunteer hours. So there was a question in here. It says, what would count as an EMG volunteer hour for this? Just photography or the time spent doing the data management as well? So yeah, all of that will count, but we have condensed this. We've condensed this to 1 volunteer hour reported or approved per plant species. We figure you're going to be taking multiple photos of that plant species. And so 1 volunteer hour approved per plant species. And this is the first guideline. And so this includes time for travel, computer work, and taking the photos. Original photos only, of course. And you can use photos that maybe you've taken previously of native species. And I think I would also make a point here, and Paul and Eric can say more about this at some point, but you certainly want you to visit the list that are included on their project website to be sure that you are capturing photos that they need for the herbarium. And so maybe they could comment more about this, but certainly those photos can come from your demo gardens, your home gardens. Those are all accepted as long as they're native or naturalized species that are part of what they are looking for on this project.
I will interject that if you looked at our species pages and say, gee, I have a photo that's so much better, please send it to us.
Absolutely, yeah.
Because we may go ahead and replace the lesser photo with the better one, even though it's not on the hunt list. But we are obviously most interested in getting things from the hunting lists.
Yeah. So also, if you can go to the next slide there, Karen, for me, thank you. How do you report these? We report these in Purdue EMG Manager, which is our online volunteer management system. And under, as you can see there, I've got a screenshot of the volunteer hours reporting page. If you go to the category, there is a state category called Citizen Scientist Projects, and there's a subcategory, IU Herbarium Photo Scavenger Hunt. We please ask you to submit your activities there under those categories, and then put in the description what you did. You know, you took a photo of this species for the IU herbarium. You could even give as much detail as you think your coordinator needs to know that you what activity you performed. And of course, all of your hours will be reviewed by your EMG coordinator. If you wanted to submit hours for taking several species photos all in one entry, that would be fine too, but please check that out with your coordinator to be sure that is okay with them. But I know it can be very time consuming to report these hours. We want that to be more of a burden than necessary. So we want to try to make it as easy as possible, but we also would like to maybe track volunteer hours and that are contributed towards this project in case we need to report that at another time. This will give us an easy way to do that and run reports from our system. So that's what we are recommending as far as that's concerned. So there is a couple more questions here. Do you only want those species on the hunting lists?
Right. Now, yeah, that's what I was telling that if you see on a species page that you say, I've got a much better image, please do send it. Or if you say,
Right.
gee, this one I know to be rather unusual in the state, that would be useful as a photographic observation. So we would still love to know if you have something from your region that is known to be a fairly uncommon species.
Great, great. And do you want photos in a particular format? I believe there's some instructions there as well,
Mhm.
but maybe you could speak to that one more time.
Yeah, I think that everyone that has submitted thus far is using the JPEG format. That's very easy for us to handle. Um, I've used TIFF, and those tend to be a little bit awkward because of the memory, the size of the files. So JPEG is the easiest for us to handle.
Great, great. Thank you. So,
But if people have earlier photographs that are in TIFF or in other formats from previous seasons when they've been out photographing things, just get in touch with us. Send us one. We're pretty good at working with a variety of different formats and stuff. And so we can do the manipulations on our end.
great, thank you. So I think, in summary here, the Purdue Extension Master Gardener program, and really this isn't, this, as you saw from the collaborators, this isn't isolated to just our program. They're not just asking us to do this, to take these photos and supplement their collection of photos in the IU Herbarium digitized collection, but it is a public project. And so this actually went out as a news release to the public, and so we are just happy to be a collaborator and give some volunteer credit to our volunteers who want to participate in taking and supplying photos of these natural and native species that are missing from your collection to enhance this identification and educational tool that could be used by botanists and gardeners and professionals everywhere. So we're glad to be a part of that. I just kind of wanted to give a little summary, and so would invite Eric or others to chime in on that too.
Right. And to follow up on John's point there, when we went through and digitized the IU Herbarium, because we have Charles Deem's collection, which was the basis of his 1940 publication, The Flora of Indiana. So we have the largest and historically most significant collection. So instead of just worrying about our herbarium, we also put together things for the entire state. And we know that we don't have specimens of every single species that occurs in Indiana. And so putting together that larger Flora of Indiana project was really something that we saw as a gift that we could give to the people of Indiana. And Paul is the person who is responsible for going through and coding in all of the morphological characters that power the golden key. And here's where the final sort of capstone piece is having those good diagnostic photographs of live plants, because the truth of the matter is herbarium specimens are pressed and dried, and they are dead flat plants. And while people like Paul and I are pretty good at trying to visualize what the living plant looks like from a dead flat specimen, most people would prefer to compare living with living, which is why getting the photographs in will strengthen how easy it is for people to use the golden key. So this is really a community resource that we're helping to organize. And so everybody will benefit by having that golden key be that much easier to use, because it does have good diagnostic photographs in it. And so we are very pleased to have this as something that we can give back to the people of Indiana. And so the collaboration with the Master Gardeners program, the Indiana Native Plant Society, Indiana Academy of Sciences, these are all things that we see really as a win-win situation, not only for the short-term benefits for getting the photographs in to strengthen the online resources that we've put together, but hopefully there'll be long-term benefits as people get to know the natural areas that are right in their neighborhood, because that kind of involvement will be really important for the long-term conservation of Indiana's natural resources.
Oh, great. Thank you, Eric. Yeah, and someone asked, does this webinar count as educational training hours for Master Gardeners? Well, of course, it definitely counts. Report that to your local coordinator for approval, but yeah. So there's also another question in the chat. Do you recommend a general search and then we identify, or that we select a particular species and then look for that one?
So here's where the hunting lists are designed to try to target people's attention. They can go to the Consortium of Midwest Herbaria to get a sense of what the plant looks like, so that they've got a good search image in their mind, and to see places where it's been collected in the past. But the work that Charlie and Stella Dean did from 1905, can you imagine starting a project to try to document the county by county distribution for an entire state in 1905 before there was even mass production of automobiles? It's just mind-boggling to me, what they set out to do. So here's where their goal was to document things where if they had one specimen per county, that was good. But here's where, seeing where it's been collected, hey, there might be, 5 other places closer to you in your county where that same plant grows. And so here's where we're relying on people's local knowledge of the areas right around where they live to help think about, well, is this a wetland plant? In which case I need to go look streamside or, lake margins or, marshes or places, or is this a woodland plant? In which case, hey, I need to take a nice stroll and admire the spring wildflowers and try to find the few that we don't have good photographs of yet. So it requires people to do a little bit of their homework to try to think about things. But as we said, here's where, if you've looked at the species that are in flower in your area and the photographs that we already have, if you think you can do better than the best photographs that we've got up there, we're interested in those as well, because the better we can get the photo documentation, the easier it will be for everyone. Great,
thank you. Well, there's another question from a familiar listener. Is there a preferred browser for using the Midwest Herbaria entries? And the photos seem to be cut off on her screen on a Mac, and it happens in both Firefox and Chrome. So
I think the problem is I have my Mac open, and initially, yes, you see the banner and a few photos, and then the others are cut off, but at the very bottom is a click to display 100 initial images. And when you click on that, then you can scroll that whole screen down and see all the pictures that are further down in the screen. So you have to find that that item at the very bottom. Oh, I got to thank you. That helps. Good.
Yeah, great.
And also I'll mention that for any photograph, you can click on it and it opens up the page for that photograph. So if there's any associated information, you'll be able to see that information. And there's usually a link to see, the largest possible version, and that largest possible version depends on what somebody actually submitted. So here's where the Symbiota platform that we use is incredibly powerful. And so we haven't gone through all of the features of it, but just, here's where you can't hurt it. So feel free to click around and see what happens when you do things. But basically, all of the information that is there, you can download in one form or another. And so you can download individual pictures, images of herbarium specimens, spreadsheets, maps, all sorts of things.
Great, thank you. So there was another question here. So seeing a native hasn't been documented in a particular region will be just as important even if you don't need a photo. And I believe the answer to that is yes.
Yes, that's true. Yes, it could become then a photo observation. So if that photo is sent to us and accompanied with the details about where it was photographed, when it was photographed, and importantly, that latitude and longitude found through your smartphone or Google Map, then we'd love to put that on as an additional data point within the database of specimens, but it'd be photographically vouchered rather than using the physical plant specimen.
Great, great. Thank you, Paul. So, Karen, Amy is also on, Amy Thompson. I think I've gotten all the questions here. Do you have any last comments here before we close this? We're just about at 1 hour. So I don't know if Karen or Amy have any additional.
Nothing from me, John. Thank you. And thank you, Eric and Paul, for coming and or being here wherever we are, and sharing this. Look forward to sharing this with my master gardeners that couldn't be on. So this will be recorded and shared out through our networks.
So one final thing that I'll mention is that each month we will update the hunt lists as the seasons progress. So what we have available right now are the things through April, but when we get to the end of March, we'll update things leading into May. So there'll always be a little bit of sort of advanced notification of what's coming up based on the flowering times that we have recorded for each of the Indiana species.
Great, thank you. Well, if there are no other questions or anything for us today, I want to thank our guests today. Thank you to Paul and thank you to Eric for including us into this project. Eric and I have talked numerous times on the phone about this and the Master Gardener's involvement, and so I'm glad we're able to move forward with this. And thank you to Karen and Amy for their help in organizing this webinar and the information for Master Gardeners, Purdue Extension Master Gardeners. And with that, we hope you all have a great day, and happy hunting, I guess, is the final word here. And we thank you for your assistance out there.
Absolutely. Thanks for organizing this, John.
Yeah,
Oh, you're
thank you very
welcome.
much.
You're welcome.
Okay. Bye all.